Well, thank you. It's indeed a pleasure to be here and talk to you about the work we're doing to try and understand the genetic code from a number of different approaches, from designing life to trying to recreate it. So first let me mention I represent three different organizations. This is the Venture Institute, again on the UCSD campus, so this is the UC San Diego Symposium this morning. It's the first carbon-neutral research building on the planet. It generates all its own energy from sunlight and has a beautiful view of the ocean. We've been trying to understand going from genotype to predicting a phenotype. And a prevailing question is, can the genetic code predict your current phenotype, and can we predict the future? And also, can we rewrite that code, which the next speaker will address with the wonderful discovery of CRISPRs. So when we first sequenced genomes, we converted the biological 4-digit code into ones and zeros in the computer. And we've been filling those databases ever since sequencing of very large numbers of cDNAs, which led to the first genome in 1995. In fact, within the first year, we did 3 genomes, Haemophilus, a small mycoplasm, and then the first archaea with the late Carl Woese. And this allowed the first comparative genomics and created a number of new questions about, does DNA contain all this information for cellular life? These genomes were all very different from each other. And could we come up with a minimal gene set that actually defined life? And that was a challenge. We decided the only way to do it was to try and synthesize life from basically from scratch from the genetic code. So we decided to start with the ones and zeros in the computer and 4 bottles of chemicals and recreate the genetic code to see if we could reestablish what we had in the databases. We started with a simple virus, the first DNA virus that was sequenced by Sanger and colleagues, FIX174. It's a little over 5,000 letters of the genetic code. But the DNA synthesizers are error-prone. It's an N-1 situation. The longer the piece of DNA, the more errors. So we had to develop new approaches for error correction. But we went from the ones and zeros in the computer to chemically making this piece of DNA that we injected into E. coli. E. coli immediately recognized this as normal DNA, started reading it, making proteins. The proteins self-assembled to make the virus, and after a certain accumulation of viruses, the cells burst open and the virus was able to infect additional cells. So we call this a situation where the software builds its own hardware. We just put in a piece of chemical software, ended up with these viral particles that could then infect other E. coli cells and make millions to billions of copies of themselves. Now, we wanted to make an entire chromosome for a self-replicating cell, but we knew if we could make viral-sized pieces accurately, we might be able to accumulate a lot of these, link them together in an assembly to get to the chromosome. And that's basically what we did, but it took a lot longer than we thought. We had to develop all new chemistry tools to be able to write larger and larger pieces of DNA and then to assemble those together. This accumulated in 2010 with making this 1.1 million base pair genome from Mycoplasma mycoides. It was made by starting with 1,000 base pair pieces, putting 10 of those together to get 10,000 base pairs, putting 10 of those together to get 100,000 base pair pieces, which we simply injected into yeast using the homologous recombination that assembled these into the entire chromosome. We had to learn how to isolate the bacterial chromosome from a eukaryote and then work on how to boot that up. So we had 2 teams, one the chemistry team and the other the biological team on genome transplantation. So the team was Carol Latigue, a French postdoc, and John Glass, who headed the team. And they probably did thousands and thousands of trial and error experiments because you cannot pipette entire chromosomes without shearing them. We have to isolate them in gel and then do all the chemistry in gels and then transform those into a recipient cell. So we have this very simple movie showing what happens when we put the synthetic bacterial chromosome in a recipient cell. We start, as soon as it enters the cell, we have a cell that's the phenotype and body of one species, and there's two different sets of genetic software. We designed the system, and my colleague in this work, Ham Smith, who is the co-discoverer of restriction enzymes that yielded his Nobel Prize in 1978, we designed it so there was an asymmetry. So as soon as some of the proteins started being made, early ones were restriction enzymes that recognized the chromosome in the cell as foreign DNA and chewed it up. So now we have the body of one species and the DNA software of the other. In a very short time, we had these new cells that are completely self-replicating. And when we interrogated them, every molecule in the cell was based on the new DNA code that we put in. There wasn't anything left from the original species. So we call this a situation where, again, the software builds its own hardware. We had to develop a new code so that we could mark these cells as synthetic. We have a code that we can write the entire English language with numbers and punctuation with a new biological code that puts in frequent stop codons. And this code contained the instructions for decoding it, a URL to send an email to it if you were able to decode it, the names of 40 scientists that contributed to the work, the names of the institutions, and then I added 3 quotations from the literature. The first one from James Joyce, to live, to err, to fall, to triumph, to recreate life out of life. The second from Oppenheimer's biography, American Prometheus, that he heard from an early teacher, say things not as they are, but as they might be. And the third, which probably is the most important, from Richard Feynman, what I cannot build, I cannot understand. After this all became public, we received a letter from James Joyce, a state attorney, asking if we had permission to use his quotation in the genome. In U.S. law, you don't have to worry about permissions for anything less than a paragraph. And then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. And finally, he sent from the Caltech archives a photograph of Feynman's blackboard with the original quotation, what I cannot create, I do not understand. We think one of his biographers changed that to the quotation you find on the literature. We've changed the genome now to reflect the new quotation. And you'll see in a minute why this was so critical. I think all this work proved and helped confirm that life is a DNA software system, and that if you change that software, you change the species. Now, mechanisms have developed throughout biology to keep this from happening very often, or you would have a nice sushi dinner and you would start to grow gills because you incorporated the fish DNA into your own. Bacterial cells use restriction enzymes as their protection. But when there's a mismatch of these enzymes, as you saw, you can add an entire new set of chromosomes to a species, adding millions of new data points in a single event. So our goal was then, after we did this control experiment, to see if we could actually design a new species in the computer. So we developed computer software to design new biological software. We actually held a contest within both the Inventor Institute and Synthetic Genomics to see who could design a minimal genome that would result in a living cell. And we actually thought this was going to be relatively straightforward, because ever since '95, when we published the first 2 genomes, there's been study after study in the literature listing what people assumed was a minimal gene set for life. It turns out none of the designs worked. And to get to this stage, it took us almost 4 more years of this actual minimal genome. And we had to add back genes until we could get a living cell again. So we had this design, build, test cycle. And so imagine doing this. We had to resynthesize the genome every time. But our tools kept getting better and better, and we now have robots that automatically build the large pieces of DNA. We went through multiple major cycles. We ended up with our synthetic version 3.0, which has only 473 genes, so it makes it the smallest genome of any self-replicating organism thus far known to science. But the most surprising part, we also made a synthetic RNA, so it would be identified as a unique cell by those who follow 16S RNA. But the most unique aspect of this new self-replicating cell is that about a third of the genes, 149 genes, were completely of unknown biological function. All we knew is that if we didn't have those genes present, we could not get a living self-replicating cell. We assumed maybe 5%, not up to 30%. And if we hadn't followed Feynman's approach of trying to build it, we wouldn't have known that we were missing about a third of essential biology. Now, any cell type, if we start with an archaea, we start with anything else, would have its own unique set of at least 30% of its genes being of new unknown function. And when we look at these phylogenetically, these unknown functional genes are highly conserved throughout the evolutionary tree. We also decided to reorder the genome, because if life's going to be part of a design process, you'd like to have an engineering approach where you had cassettes of the different gene systems. So we, using another computer term, called it defragmentation, where we can reorder these. And just to show you how complicated it is, this is just looking at all the rearrangements on resynthesis to get even 1/8 of the genome reordered. I think the most surprising thing is we can do this and results in a living cell without any change in the biological function. So what we see in genomes is the results of billions of years of random events, and we can reorder them and get them in a highly ordered function, which will make the next levels of design much easier. We can now, we think, recapitulate, as scientists in the 17th and 18th century proposed, all of biology by adding genes back to this minimal cell. This helps put in context our 22,000 genes in the human genome that we know even less than 2/3 of in terms of any one of the biological functions. The same techniques that led to the first genome of using computational tools to assemble all these pieces of DNA led to the first human genome in 2000 and this publication in 2001. And a few years later, the first complete genome separated into complete haplotypes of the 2 parental chromosomes. This first genome cost $100 million and took 9 months and was not an event that was going to change the practice of medicine or even our understanding of individual biology. Fortunately, what's happened over the last 15 years is a major change in technology. Very little change in our understanding of the genome because we only had a few genomes to work with. but sequencing costs got below $2,000 a genome instead of $100 million. And most importantly, computational power has changed substantially. In 1999, we spent $50 million building the third largest civilian computer in the world, which was 1.5 teraflops. Many of you know you can buy a card for your PC for about $100 that does a teraflop versus computing. And we've gone from major computational centers to distributed computing with cloud computing. The third component that we're waiting for was advances in machine learning, because the amount of data we're dealing with is much too vast for mere mortals to try and understand. So a little over 2 years ago, we formed Human Longevity. This is our main building in La Jolla. We also have a site in Mountain View, which is our machine learning team, and a site in Singapore. We started with the Illumina X10 sequencing large numbers of genomes. A little over 30,000 have been done to date, but that results in close to 4 petabytes of just ACs, Gs, and Ts in the computer. To put that in context, just a few years ago, indexing the entire internet was only 12 petabytes. With our goal of over 1 million genomes, just the sequence data alone is going to be the largest data set that anybody's compiled. And just, the definition of sequencing genomes is now become a sort of like the Republican laws in the US, where all data is fungible. There was a paper recently published in Nature with the title of The Sequence of 1,000 Human Genomes. It turns out they were only done to covering less than 30% of each of the genomes. At HLI, we sequence genomes to 30x coverage, which is the only way to get statistical coverage of about 10 sequences for every base pair, which gives us highly accurate data and coverage of the genome. Even so, human genomes are still not completely sequenced. I don't know if you can see these gray areas. That's where there's less sequence coverage. These are around the highly repetitive areas of centromeres and telomeres and other repetitive areas. But in the reproducible genome, you can see the false positive and false negative rates are extremely low, resulting in extremely accurate data. Now, even before we reached 10,000 genomes, we saturated all the common variants in the entire human population. And this is where if you've got a 23andMe chip or another gene chip data, they measured just the first couple of bars on the right, the common variants that most of us share. These are not the causes of disease or even any of our uniqueness. Now, if we sequence anybody's genome, we add about 8,000 new, extremely rare variants that only show up mostly at once in our entire database. These are the ones that are highly significant in terms of causes of diseases or other genetic traits. In fact, now we can do calculations that we could only have dreamed of a few years ago, where we can look at billions of data points to understand the structure of the entire human genome. And what we have found is there are invariant portions of the genome that if there's a sequence variation at that site, it's incompatible with life. These are quite often at intron-exon junctions, but a number of other unique places. And even we can take all the transmembrane proteins together and look at their functions. And perhaps not surprisingly, the part of the protein that spans the lipid membrane is not mutable in a way that's compatible with biological function. It's not surprising if you get a charged amino acid there, the protein couldn't stay in the membrane. Both the intercellular and extracellular part can take high levels of mutation. Now, I think one of the most important discoveries the team has made recently is certain mutations and truncations in genes disappear with age. So, in other words, they're incompatible with long life, and you don't find them in older populations. This is the average curve if you were born in 1900. The blue line, you can see that getting to 50 was a big deal. If you're born in 1950, the yellow line, it's a nice shift. And if you're born as recently as 2010 is the red line. We're trying to make the shift to at least move this curve out, not necessarily where you fall off a cliff at age 100. We don't know what the true biological limit is as we find these variations. But one of the biggest surprises to me is our genetic code is constantly changing. If you'd asked me 15 years ago, I would have said you get your genome sequenced once, and unless you have cancer, that's all you need to worry about. Now we can very accurately estimate your age just from changes in the genetic code. So it's not going to be simple to solve the aging problem, but these unique gene truncations that only occur as heterozygotes yet result in early death are going to be an amazing set of new targets. We don't need huge pedigrees and huge populations now to understand disease. We can sequence from a single patient the genome and understand the changes associated with that disease. This is an unusual disease, Delleman syndrome, that only occurs in, thus far, about 30 individuals in the entire human population. If you can see the MRI, the brain is highly scrambled. It's actually more amazing to me. This young boy has any appearance of normal function. His body is covered with these small tumors. We had the ability to get the DNA from his parents as well, so it's called a trio when you have the two parents and the child. And it was very interesting because just looking at the parents' genomes, these solid color areas of black and solid orange is where his two parents had the identical genetic code over these long regions. That tells us immediately that the parents are very closely related to each other and a result of a lot of interbreeding. They were in fact second cousins, but they're from a small village in Mexico with a lot of inbreeding. But any geneticist can tell you if you have the identical sequence in both parents, it means extremely rare alleles then get expressed. Looking at the boy's genome, we found a number of unusual genes associated with neuronal development that were in fact mutated. And one very unusual one in combination with that, and that's the neurofibromatosis 1 gene. Many of you probably know it as the elephant man's gene. It was changed as well. But this is where we can now find mechanisms of inheritance. Having both parents' genome, it turns out this mutation in the NF1 gene only occurred in the child, which means it was a spontaneous mutation. So you can't rely just on looking at your parents to understand your own genetics. We all get several hundred to several thousand spontaneous mutations that make us totally unique and even different from our parents. But now just from a single case, anybody trying to diagnose Delleman syndrome has the genome for comparison. One of the areas clinically we're finding the most interesting for applying some of these tools is with cancer. At HLI, we have a comprehensive cancer analysis where we sequence the entire genome of the patient, the entire genome to 90x coverage of the tumor. We sequence the RNA in the tumor. We isolate T cells from the tumor and sequence the entire T cell population. We sequence the viral population. And we also do liquid biopsies looking for circulating DNA. And sometimes we also do the metabolome. This results in an analysis that gives a lot of information. And here's an early case of HPV-16 caused head and neck cancer, which is a new, somewhat epidemic in men, usually starting in their 40s and 50s, that it's not clear yet whether the vaccine given to young boys and girls will work as a cancer preventative. But just on sequencing this man's genome, we found he had several mutated oncogenes, which put him at high risk for cancer. Genes from his immune system that indicated he was probably immune suppressed, which he was. And from his microbiome, we found HPV-16 present. Just that unique combination would probably say this individual would be more when he got cancer, not if he did. On sequencing the genome, we found 25,000 mutations. So doing both the patient's germline and the tumor, you get the mutational burden of the tumor. In this case, 25,000, which is sort of a modest number. 315 of those mutations changed protein structures. So the term for this is neoantigens. We also found 4 places where the HPV virus inserted into the genome, disrupting a gene function. So we're able to do pathway analysis, not only looking at the mutated genes, but looking at other druggable targets in that same pathway. And by sequencing the entire T-cell repertoire, we can tell whether the patient had mounted an immune response against any of these neoantigens. Now, this should hopefully make it clear to you, if it hasn't been before, why your immune system can detect cancers. The mutations create new proteins that aren't normal human proteins. Your immune system, if it's intact, recognizes these as non-self, and your T cells normally will kill these cells and rid them from your system. We are constantly ridding ourselves of tumor cells. If you go out in the California sun for about an hour, you accumulate about 10,000 mutations in your skin cell genome. Fortunately, we're constantly replacing our skin roughly every 2 weeks. That dust in your house, that's you, from you're constantly shedding your skin. But other cells don't get replaced like that, and we can accumulate mutations until they become cancerous. We have a number of new approaches from the druggable targets, but with these neoantigens, we can take a number of different approaches. One with an HPV-16 caused tumor in a patient that's been completely unresponsive to any of the chemotherapies. We actually isolated the T cells from the tumor and then made peptides from the neoantigens to select the T cell populations that would attack the tumor by recognizing those neoantigens. We grew them up and as a population gave them back to the patient, and they've been pretty effective at dramatically shrinking his tumor thus far. But for a longer term approach, we're developing RNA vaccines against these same neoantigens that we're testing both to see if they can create immune response against the tumor in combination with other immune approaches, but even act as a preventative. For example, with prostate cancer, we see precancerous lesions with accumulated mutations that have not yet yielded a cancer cell. We should be able to make RNA vaccines at that stage, but it's early on, and that's some of the speculation. Now, one of the things that was critical to us was not just to have genome sequence. While that's useful for some of the analyses I showed you, the only way that we see to interpret the genome is to have very large numbers of genomes, because all of us in the human population differ only about 3% from each other. But we need phenotype and health data for comparison. So at HLI, we started the health nucleus as a way to get this phenotype data and to try and change the practice of medicine from what it is currently of reactive. You get symptoms and you go see your physician. They try and work out what those symptoms are related to and come up with a treatment, to a proactive, a preventative approach. And I'll show you some examples of this. Now, if you're between 50 and 74 in the US and you're a male, 30% of you will never reach the age of 74. If you're a female, 20% of you will never reach the age of 74. And hopefully these graphs show the main reasons why. In males, cancer is a little over a third of the cause, even slightly greater in females, and cardiovascular disease is approximately the other third. So with cancer and heart disease, if we could do early predictions, early detection, it would change things quite dramatically. And the other third of diseases we're also looking at. We take a number of imaging approaches. This is a GE3T MRI that we're using with some unique research protocols that aren't yet generally available. One of the advances in the last 2 years is this new restriction spectrum imaging that simply looks at the water molecule differences, for example, in tumor cells versus normal cells, or blood vessels versus surrounding tissues. And so without any contrast media in a short time in the MRI, we can get pretty stunning images. And we can diagnose cancer without contrast media and without biopsy. So this was a study recently published by our key radiologist, David Caro at UCSD, where the cancer cells show up in this bright yellow images, basically a light where there's cancer. And when the prostates were removed, the regions corresponded to exactly what the pathology showed. This is a radical difference from measuring PSA levels and then getting random biopsies, hoping to identify cancer. It's even more dramatic with images like this. 8 minutes in the MRI with no contrast, we can get a complete map of your brain vasculature. Why is this important? Well, if your carotid arteries are getting blocked, or in a case like this, a young woman with a brain aneurysm, we can detect things early on when they're totally preventable in terms of the outcomes. Turns out 1 in 50 in the human population have aneurysms, and they're usually detected by people having sudden death, or in the case of brain, they can have a severe stroke if it doesn't kill them. Just interventional radiology can simply put a coil in and block that from being a problem. The previous speaker mentioned looking at the hippocampus and looking at disease. We now have algorithms developed by our colleagues at UCSD that give us quantitative data of 20 different brain volume regions where we can detect changes in as little as 3 months. Now, the hippocampus is this yellow area in a normal brain. You can see it's quite large. We get the volume of that very precisely, but you don't need that to see the tremendous difference here where the hippocampus is shrunk and there's just open volume in the area where the hippocampus was. It's pretty easy to diagnose dementia with that. All the people that come to the health nucleus think they're healthy and disease-free. We're finding significant disease in about 40% of them. Everything from something like a cavernoma, which is a vascular tumor in the brain. Here's another one, if you can see this area at the top of the brain. It's another vascular type tumor. You wouldn't want to be a football player and get constantly hit on the head with that. It would certainly bleed. Here's a case where the new imaging techniques make a huge difference. This was a 56-year-old man who came in who was going through the health nucleus, was going on a world tour vacation the next day, or so he thought. We discovered this 5-centimeter tumor right under his breastbone. With the differential image, you can see on the right it showed up as a cancerous tumor, essentially lighting up like a light bulb. Within a week, using the MRI image data, it was robotically removed, and the tumor had just started to penetrate the surrounding tissue. So we caught this at stage 1. So it was an individual who thought he was healthy for 1 week, knew he had cancer, and is now cancer-free. Had he waited until this grew a little bit larger to cause symptoms, he would have had to go through some extensive chemotherapy and would probably have about 2 years to live. So that's a dramatic difference in longevity. And this is very different than what we get from all the tumors in our comprehensive cancer program, where they're usually diagnosed at stage 4. For example, right now, we have 3 women in their 30s with stage 4 colon cancer. It's so much more satisfying when we find these tumors at stage 0 or stage 1. We can also diagnose metabolic disease right in the MRI because you can quantitatively measure the amount of liver fat. Normal is 4% or less. And we're finding people with up to 38% liver fat that will certainly have liver transplants in their future, being completely unaware of this. This is a case of the non-alcohol related one called NASH that can be diagnosed right off of these images. We have a little patch that we give clients to wear. It's about the size of a Band-Aid. You wear it in your chest for 2 weeks, and it records your EKG 24 hours a day for 2 weeks. And we've found now in 5 people life-saving data is 3 examples where people had episodic AFib for up to 7 or 8 hours a day, being completely unaware of it. You probably know that's one of the major causes of stroke from coagulation in the vibrating atria. They were simply put on anticoagulant therapy. We had 2 individuals with complete heart block where their heart rate went down to 20 beats a minute, and we're somehow unaware of that. Part of the exciting discovery phase now is taking what we find from the clinical observations and matching those up with the genome. For example, polycystic kidney disease, we know the gene for that, and we can verify whether this is just a statistical probability or what rate we find these. We found a number of, using 4D echocardiograms, people with distended aortas, and we found a single gene duplication that seems to correlate with those. The last thing I want to tell you about is how we're applying machine learning to try and understand new features. You probably all have used Google Translate at some time or another. We used it yesterday with our driver to communicate between English and Japanese. Franz Ock is the guy who designed that at Google. I hired him to head up the team, and the first challenge I gave him was to see if we could predict a photograph of your face from your genome. But in your genome report that we give people, it's over 500 pages now, we can actually predict your genetic height, your BMI, your eye color, your skin color, your hair texture, and a photograph of you. We can also predict your age. Men and women, as we age, we lose sex chromosomes. We lose Y chromosomes if you're a male, and X chromosomes if you're a female. From our sequencing, we get a very accurate measurement of the telomere length, and obviously that correlates with age, probably not in a causative fashion, but it's a good measure. We can also measure mitochondrial sequence changes. So we wanted to try it with face prediction, so we started a clinical trial. We recruited 1,000 subjects and took 3D photographs of their face. We sequenced their genome and measured a number of other parameters. We're also trying to predict voice from the genome. But from a digital voice recording, we can tell very accurately your age, your sex, and interestingly, your height, which is an indication of your volume. That's just a digital recording you could even do over a telephone. With these images, we built algorithms with machine learning that allow us to do some increasingly accurate predictions. These are photographs of one woman with a tremendous diversity in her ethnicity. The top is the 3D photos, and the bottom is the straight computer predictions just from the ACs, Gs, and Ts from her genetic code. 3D photographs aren't very friendly to people's faces. So for the computer predictions, we go from their actual photograph, we actually smooth them. And the computer algorithm right now predicts your face with complete symmetry. So people like their face predictions because our prediction of beauty is based on that symmetry. And also, we didn't know what age your genome would code for. It turns out it codes for just post-puberty, so maybe age 16 to 18, which we find interesting. Here's another one with the, on the far right, the computer predictions. These were with early versions. The latest version is getting much better. This was the from the smooth photo, and this was the prediction straight from the genetic code. Here's another example on a male. And this is, you can see, without having the shape of the head or the ears, and we're not including the eye color and eye shape yet graphically in these. Here's the computer prediction right from the genome. Ethnicity plays a big part in the calculation, so it's easy to get people with different ethnogeographic backgrounds. So these are starting to get more and more accurate. From your genome, aside from all the parameters I told you, we also get your blood type and a very accurate HLA type. So we get a lot of information just straight from the code. If you go through the health nucleus, we put all this together. It's a lot of data, but we also take 3D photos of people's whole body, and we make avatars, and we give you all this data back on an iPad, and the avatar helps walk you through your data to explain some of this complexity. We're early on. We're trying to accumulate at least 1 million records by 2020. We've done deals with pharmaceutical companies, including the latest with AstraZeneca, to do 500,000 genomes over the next 8 to 10 years. With the phenotype data, this is going to feed our machine learning algorithms and give us a number of new exciting predictions. So thank you very much.